We will have one speaker joining remotely, Stasha, who was not able to participate. Hello, Stasha.
Hi, everyone.
Okay, it works. You look very beautiful on this big screen. You don't see yourself.
Thank you. You too, guys. Very professional and beautiful.
So since I think you're the only person. About yourself and what you do, and please.
Firstly, I'm so sad that I wasn't able to join in person. There are many perks of being Serbian, but one of them isn't to have a visa for Ireland. So that was the reason why I'm not there with you. My name is Stasha Stankovic. I am a bioinformatician and a geneticist. I did my PhD at the University of Cambridge, where I studied genetics of ovarian aging and menopause. And I'm also a strategic consultant at AstraZeneca, where I shape how we discover novel drugs using human omics data and artificial intelligence. I'm also founder of a startup which was basically aiming to commercialize my PhD research into more novel therapeutic opportunities in this space, and I'll be happy to tell you more about this in the panel.
Thank you, Stasha, so much. And maybe Jennifer as well, because to introduce yourself, I think the other speakers had a chance. So please tell us what you do.
So I am from Toronto, Canada. I'll extend a little more than what I was able to share yesterday. 20 years in medical doctor, 20 years in practice. I run one of the largest concierge, holistic, regenerative women's health practices, and for about a decade, helped to run the largest hospital-based menopause clinic in Canada, and worked closely with what may still be the only clinic focused on POI in North America. So there was a lot of synergy in working with a cohort that we heard quite a bit about with Noon's talk. So I have sort of merged the role in functional medicine and longevity medicine with a female-centric approach.
Thank you so much. And I mean, the topic of our panel is combating reproductive aging. And with that being said, I think we already heard from everyone that the main problem is that we are being born with finite number of oocytes that are decreasing in their quality and quantity over time. And I would love to actually give some a spotlight as well to our ability currently to predict the onset of menopause. One thing that I think Neva as well mentioned, that it's also based on that number where we start, and that could predict the onset of menopause, but maybe genetic as well. Genetic research can also tell us something. So Stasha, can you tell us a bit about current abilities for us to predict the onset? Yeah.
Absolutely. I think maybe firstly to start and kind of highlight that the reason that we have such little innovation in this space is we didn't really understand what are the biological mechanisms behind ovarian aging. So why do our ovaries age 3 to 5 times faster than any other organ in the body? And if you don't understand the root cause of why something is happening, you are not able to innovate, both in the kind of predictive, diagnostic, but also therapeutic space. So why the genetic approach was so revolutionary is that for the first time, we were actually able to start understanding what are those genetic risk factors that are driving ovarian function and longevity. And if you think about clinical setting and what we are seeing now in terms of the prediction, there are certain biomarkers or sex hormone that we are currently utilizing to assess someone's fertility. These include follicle-stimulating hormone, luteinizing hormone, anti-Mullerian hormone, etc. But the main problem with those sex hormones is that they don't have long-term predictive capability. So what does that mean in terms of reproductive aging and in general planning the fertility journey? We know that fertility or infertility actually kicks in 10 years before menopause. So there is this lag period between the major infertility and your official entrance into menopausal state. And that lag period is basically characterized by very much big difficulty of conceiving and having children. And I'm sure the other speakers covered that before. So the problem with the current biomarkers is that they cannot predict when that perimenopausal and menopausal transition will take place, and they can only assess what your ovarian reserve looks like at the moment. But as this is a very volatile process, we do need to understand and know when that transition will take place so that we can enforce and inform women how they should plan their fertility journey and family planning in general. So the power of genetics is that we are born with it. It's your barcode. It's a lottery that you're born with, and depending on what is the genetic combination that you carry, that will determine your menopausal timing. So we've studied more than 200,000 women. We try to understand what are those parts of DNA that actually predispose them to be early menopausal versus maybe candidates for primary ovarian insufficiency, or to have an average age of menopause. We discovered about 300 genetic factors that are actually guiding this process of reproductive aging and menopause. And what we can do now is we can actually put all those genetic risk factors in a single genetic test and apply it with the clinical potential to try predicting someone's menopausal timing. At the moment, we have about 70% capability to differentiate women who will have normal menopause versus women who are candidates for early menopause and POI, and therefore enforce them and inform them how their reproductive longevity might look like, so they are at least informed whether they can delay their first age of being mothers, or they should kind of reshape their family planning around that information, because they will know if they are, for example, candidates for early menopause. Obviously, genetics is not the only factor that is guiding ovarian aging and longevity. So we will have to combine genetics with other factors, including sex hormones and counting the number of eggs in the ovary, in order to have a very certain and powerful prediction of the menopausal timing. But this is an incredible step forward when it comes to having a really informative and powerful test in this field.
Thank you, Stasha. I mean, this looks very promising. Do we know, I mean, how far are we actually to have this in clinical practice? Maybe that's like how close we are to have it being applied in clinical practice?
So at the moment, the genetic factors that we've discovered are only coming from European women. We know there is a different genetic makeup when you look at menopausal timing of European women versus African versus Asian. So if we want to increase the power of our test, of our potential test, we are now starting to incorporate the data on multi-ancestry population. But as I said, after that step, the next step would be to really carefully think how we can implement and combine genetics with all these other factors that determine your ovarian aging and longevity. So it could be 2 years from now, it could be more than 2 years, but we've already did the major step. So now it's just about hitting those additional 10 to 15% to reach the power to have a really confident solution in our clinical practice.
I think the clinical practice is definitely being excited by about this fact. So the sooner the better. Yeah. Jennifer, you wanted to add something?
I was just going to say, Sasha, it's so great to connect with you. And we're actually doing this. So I want to learn more about your work. So we do this presently in clinical practice. And maybe I can just expand on that incredible explanation of Sasha's work. We do whole genome sequencing. We have AI-powered sort of third-party analyses that look at propensities around fertility and early menopause. And we combine that with serial clinical monitoring over time, because we know biologic and chronologic age start to diverge significantly in the late 30s and by the early 40s in women, such that, typically testing done, both follicular and luteal phase testing done over a few cycles, several months apart, give high predictive value for women. And very interesting population that I treat that I just want to speak about, because amongst high performers, this is a frequent occurrence, is female athletes. And we know of the athletic amenorrheic syndrome, and particularly these days with PEDs, performance-enhancing drugs, and hormones, and all the things some of these female athletes are doing to themselves, we are seeing a foreclosing of their fertility window. So I do a lot of this with that cohort. And that cohort, they're in their 20s, and they look 40. Physically, and the physically and the ovary.
Sorry to interrupt. How expensive is to do it at a whole genome level?
So I'm in Canada, but we have several genomic and genetic groups that we use. And I can tell you that in the last 5 years, the whole genome sequencing has come down in price significantly. What we charge the consumer, the patient, includes third-party reports and our time to go through the whole analyses. But so I can't give you one price because it really depends on how we customize that. But at the end of the day, the diagnosis, the prognosis, and the therapeutic interventions are precise and personalized and evolve over time.
Maybe that's a good actually introduction then to a question that I wanted to actually ask Maria, because Maria, you talked a lot about also epigenetic factors that are also impacting the reproductive health and the other exposures. If you would, or whoever wants to take the question, when we look into genetic and epigenetic factors, what are usually taking the most dominant role in the onset of menopause and aging? Who has the larger impact? Maybe saying to that, if you would like to take, or somebody else, I don't know.
Sure, genetics wasn't my main focus.
Mm-hmm. Yeah.
There are some studies showing that there are epigenetic changes with reproductive age around DNA methyltransferases, around histone modifications. But apart from the genetic factors, there is obviously a role that environment plays. So I actually wanted to ask Stasha if she's taken that into consideration and how would that fit into the prediction model.
Sure.
Yeah, we have taken into account smoking. We know that women who are kind of at high risk or high intensity smokers, they tend to have menopause 2 years earlier than they would initially have. So far we've only taken into account smoking, but not other factors. I have to say that we did look at how nutrition impacts the menopausal timing, and not only in the individual, but also on the offspring. So for example, pregnant mice who were exposed to high fat, high sugar diets, their offsprings tend to have earlier age at natural menopause, or mice don't actually have menopause, but a smaller ovarian reserve than the wild type with mothers who were exposed under kind of Mediterranean/healthy diet. So we know that lifestyle does not only impact your own menopausal timing, but also of your offspring during pregnancy. But in terms of the genetic test itself, as I said, we just took into account smoking, and we're now kind of expanding that into considering other additional factors that might play a role in shaping your menopausal timing.
Can I share an insight from our POI population? I think it's so relevant and pretty profound. When we heard the concept of the supercentrarians are highly determined genetically, I would say the reverse is true for the POI and early menopause cohort, that it's predominantly determined epigenetically. And I actually found that really interesting in your talk as well. And I would just propose this is something we would see repeatedly 4 predominant epigenomic triggers or factors. One, Sasha, I think you're alluding to, is the transgenerational effects of epigenetics. And we know actually that from a transgenerational effect on the germline, it's the 2nd generation. So it's actually looking at maternal grandmotherhood, stressors like low protein diet and extreme psychological stress. And because the egg of the 1st generation is already formed in that woman, grandma, experiencing the stress, it's her granddaughter that will manifest the fertility effect of that. That's one. Second, cigarette smoking is highly toxic to the oocyte. That would be an important exposure risk. But I would say probably the most important epigenetic risk to a woman to predict an early menopause is psychological stress. We see this in events like an immigrant coming without family support or a significant illness in the family. And then last is just something interesting to think about, just surgical or health risk to physical kind of stress to the patient. And importantly, hysterectomy, ovarian sparing surgery, like removing the tube and the uterus, I mean, this is not relevant from a fertility perspective, but in a cohort I see a lot of these women that are not told by their OB-GYNs that they're having, for benign reasons, a hysterectomy. They take the tube and the uterus, they leave the ovary, they say, don't worry, you won't be rendered menopausal, but there is crosstalk. And they do experience, not POI, but typically they're in their 40s, these women, because it's after their childbearing years, but they do experience early menopause. So those are 4 factors, all we can think of those epigenetically, but not genetically based.
Thank you for sharing. Maybe just because now conversation went in this direction, Niamh, if you could also maybe share from your practice, because this is quite interesting, right, for patients who have kept the ovaries, but maybe removed other parts of reproductive system, and any effect that you observed in clinical practice. And what is the hypothesis at the end? How is that all orchestrated together? Because it seems that just focusing on ovary is not enough. So.
No, it's not. I suppose we know that the fallopian tube and the ovary have a shared vascular supply. Yeah. While the ovary is not entirely dependent on the blood supply that's shared with the ovary, it mostly comes from the pedicle that I showed in my talk there. The vascular petals are all in the suspensory ligament. But we actually now do counsel women that there will be an increased risk of an earlier menopause with removing the tubes, because it's just the way it goes. But then there's also the added benefit of removing tubes, because we believe that ovarian cancer comes from these stick lesions that come from the end of the fallopian tubes. So it's kind of, you have to balance it out. Why are you having your reductive surgery? Are you having it because you have a risk of ovarian cancer, but you want to keep your ovaries for a little bit longer, but you want to get rid of the precursor lesion? So it is complicated, but certainly there is a risk of an earlier menopause.
I mean, I'm also suspecting that there is going to be a time where we still need to really look into what substances are these organs producing, because maybe it's not just estrogen and not just progesterone. And there are so many other aspects of the whole system affecting our whole health and orchestrating everything that is happening. I think we even had a talk before, right, what the effect of estrogen on our immune system and the quality of chronic. So I think there's so much more to discover on what are these cells producing. And I mean, maybe in parallel, looking into animal models. So maybe looking into what are these extraordinary animals, naked mole rats, doing better than humans and staying so healthy. And have you any observances in their genetic code, epigenetic factors? How are they?
Well, so randomly enough, we share a lot with the naked mole rat from the point of view of expression of the genes and conservation of the genes. What is really different is like the clock. So we stop like expressing some of these genes way before than this animal. Mm-hmm. So I think that the important part here is to learn how to control that. Mm-hmm. And actually those genes that for example, we are analyzing and seeing how they conserve in human and mouse or doing transgenic animals show that tweaking those ones, you can extend the lifespan of the ovary in other species, including mouse. So the other important part here is that I like to say that the ovary is not just like a factory of babies. So that means that it's producing many other things that we don't know. Some of them we know, but actually it's a big black box that we need to analyze. But when it comes to the aging, we think that, okay, the ovary is done, that's it. But imagine that you have a mega combo of senescent cells, inflammation, so it's something that chronically will be there for the rest of your life. And I think that no one wants to have like a scar tissue secreting like saps or getting senescent cells and going everywhere. So I think that there are like many layers that we don't know, and we are trying to figure out how. And the naked rat and the extracellular matrix and the genes that are expressing are helping us to protect this or to move forward. Also, what I want to say is like, we talk here during the last few days, a lot of biotech companies, and you're targeting people that are going to have hypertension, people that are going to have Alzheimer's. But here we have a market that is covering 50% of the population worldwide, and we don't have anything to give them. So I know that it's kind of new, and I don't have a company, and I'm not asking for money, but think that this is a really good business. So we need to invest in this, and it's beyond having babies. It's extend the quality of the females. It's like, yeah, they live normally longer than males, but they live in this quite not great way. So
yeah, to complement that, what you're saying, I mean, if we put in a perspective, this little organ has the power to transform human body from a girl to a young woman, to grow different organs, to then transform it into a mother, to completely transform the biological function, and then again, transform body again into a normal stage. So the power of these hormones are just enormous. So if we understand better how to play with them, then I think the power is just enormous. But then coming back to what you just said, have you observed, because the benefits, right, of species that are having or not having menopause, have you observed in observing their health span of subordinates even, like any benefit? I mean, overall you share that they're in a very good health, but any particular observances of the risk of other diseases? Have they shown anything? Because we are always thinking in risk of ovarian, like cancer, breast cancer, endometriotic cancer. Have they shown any of this?
So subordinate females in the naked. Okay, let's think about that. All the animals that are not queens, or the 4 or 5 males that are active, they're like block. So imagine that you have all the population in blockers, so they never got really through puberty. So they're keeping them there. So technically the risk of cancer in the naked rat is really low, and also other diseases. So basically, for us, having naked rat in the lab is to learn how they protect them from these diseases. Now we have, like, for example, targets related to extracellular matrix in the naked rat. There's a lovely paper published from Vera Gorbanova talking about this. But when you target this in the ovary, you actually trigger a phenotype that is similar to what is happening in human or mouse. When you actually start like moving faster everything in the ovary, they get into this kind of into process of differentiation and apoptosis, and they lose the ovarian reserve. And the same when you trigger this gene in mouse. If you overexpress this one, you can protect the ovary, extend the ovarian reserve, and also it seems like it's protecting to the cancer and other stuff.
So if you would highlight the biggest potential to translate this research at the moment, would it be like in some therapeutic agents, or? Yeah.
I mean, everybody will like to say, like, let's do some kind of gene therapy. However, these genes that I mentioned are like enzymes. And the lovely part of the enzyme is that we can develop drugs. So, and also there are different pathways. So we can stimulate them or we can block them. And they're highly conserved across the species. And in fact, part of our experiments using human samples, they show that actually they're there, exactly the same, just they stopped working before. So if we can create something that is moving in that way or activating these enzymes, it's great. And we have those kind of prescription every day. If someone here has hypertension, you have ACE blockers or things like that related to angiotensin
So it would be like a blocker of these enzymes,
or
and
like
then or
inhibitors. Yeah.
inhibitors. Amazing. And then maybe looking into the back again to clinical aspects, but as well, I think Stasha, because you looked so much into human models in comparison, can we talk a little bit and touch upon, okay, the benefits? So ideally, if we get rid of menopause, because we definitely see that this is not a biological, it's not a must to happen. If we are very successful in removing it, so how would actually our health span, how will that affect our health span? And what are the benefits of it? And what are the downsides? So what are we have to be cautious about? Liv, would you like to take the question?
I think I kind of just touched on it at the end on my last slide there, and I suppose we go through menopause, and then as humans we live for another third of our life practically, albeit not well, but we do survive for some reason or another that I don't think we completely understand. I think the benefits are definitely notable because we can see the effects of estrogen, particularly on perimenopausal women. And I think they're the women that need to be supported just through that transition, because it's those fluctuations that really wreak havoc. And why it goes on longer in some women and not in others is probably a genetic element to that as well. But then beyond that, it's like I'm not sure if we should not be going through menopause. I'm not convinced. And that's probably quite controversial to say here. But I think we need to understand also more about how we age in a stage of life where we are not exposed to estrogen, and how can we bolster our well-being and our health span during that stage. And I think that's where we will need to look to science to suggest, I suppose, sensible steps to take in the clinical platform. And we need to, I think, from medicine, I think what's so nice about functional medicine and longevity medicine is that we think holistically, whereas in medicine we're quite siloed. And we do need to have collaborative efforts, particularly, say, between endocrinology on how we manage osteoporosis, cardiology, respiratory function. We're not siloed as human beings, and there needs to be that integrative effect. And I think there's more work that needs to be done in the postmenopausal stage.
And I mean, probably combining as well all the other longevity interventions, right? Protecting the DNA damage and everything, and then combining it with all the other interventions from reproductive aging. I think Stasha wanted to add something.
No, I'm absolutely with you guys, especially on kind of the topic of delaying menopause. I think it's about the balance, right? We have to highlight that early menopause is associated with the risks that were just mentioned, like osteoporosis, type 2 diabetes, etc. But then on the other side, we have that other risk of reproductive cancers associated with late menopause. So if we do want to extend menopause, and if that's a good idea, I'm also not super convinced, how can we find a balance where that shift should happen? If we do delay it, how much do we delay it? Where is the right balance of having additional exposure to estrogen that is not detrimental, but it's helpful? And I do think that, when we think about these ideas around extending reproductive longevity / delaying menopause, we have to think about patient stratification. And the reason why I'm saying this is that when we mention menopause, the topic is twofold. The first one is about fertility, which is linked to menopause, and then the second one is about the health span. And therapeutic interventions for those two groups can look very different. I think extending menopause in the group of women who are early menopausal and have primary ovarian insufficiency can be super beneficial, because it will give them some extra years to have a family and think about family planning without starting super early. Because these ladies, if they enter menopause at the age of let's say 40, that means that they are already infertile at the age of 30, and with the delay of motherhood, that just clashes all the social patterns that we are having right now in the society. And then on the other side, when we think about women who have normal average age of menopause around 50, then we're talking also about the health span. So I think the discussion is much more complex than just do we delay it and how we delay it, but kind of what are the groups of patients and individuals that we can personalize these treatments in. One of the kind of most interesting discoveries that we had from human genetics was around the fertility bit. So I can just quickly maybe explain what we've done there is from the 300 genetic factors and genes that we've discovered as responsible for determining the menopausal timing, we took two examples, CHEK1 and CHEK2 genes, famous DNA repair genes, which are associated, one if deleted or one if having a loss of function of CHEK2 with delayed menopause or later age of menopause. And then on the other side, if you have overexpression of CHEK1, you would also be going through late menopause. And that was all in humans. So we discovered this in UK Biobank, which was a massive cohort of more than 200,000 women. And then what we tried to do is we tried to mimic these mutations in mice and see whether we can observe the same effect when it comes to ovarian reserve, the size of the ovarian reserve, but then also the fertility. And not surprisingly and thankfully, we've seen the exact same phenotype in mice where these two type of mice had extended reproductive longevity by 25%, in human words, delayed menopause by 25%, and improved fertility. And what is even more interesting, if you think about therapeutic implications, when these mice went through IVF, the number of eggs that we managed to extract after the hormonal stimulation for the procedure, for the IVF procedure, was 50% higher than in wild-type mice, which is a fantastic therapeutic opportunity for women who are so-called poor responders. Poor responders are women that don't respond well to hormonal stimulation in the clinic that is necessary for the IVF procedure. There is about 40% of women in that group, which is, again, if we think about market, is a major market. So this was kind of the very first scientific explanation and evidence that delaying menopause in relation to fertility is possible, but now we have to science the hell out of that egg to understand how we can do that in humans and whether it's safe and whether it is actual therapeutic opportunity.
Thank you, Sasha. I think this is very good entering now the end of this panel. Science the hell out of it. We know that the reason why we are here where we are because this whole area has been so under-researched. And we're just now seeing the wave of research papers rising in institutes looking into this phenomenal topic that is so tied to understanding our health and what is happening. So maybe like one or two sentences to close this wonderful panel and maybe to inspire our audience. If I would ask you, where would you invest if you would be an investor? What is the most promising and exciting? I think Stasha already answered this question just right now. What is the most exciting area that she would invest in? Which type of research? Or creating options for in clinical setup. So what would you do in order to improve the future of women's health? Who wants to go first? Jennifer.
I just wanted to add, I'm going to answer your question in just adding one thing that we do know a lot about, building on what was said, and that's that just in terms of, and Sasha said so well, the two different strategies of extending the fertile window versus extending femspan, or delaying menopause, extending menopause, or managing menopause to allow women to age more successfully. Those are two very different goals. And we do know very well, this is very well established, that for the latter goal, which is probably the focus of this audience, femspan or longevity, we only need to replicate not all of ovarian function, but early follicular hormonal levels. And that is the goal of menopausal hormonal therapy. lower levels of estradiol and not cyclical surges of estrogen and progesterone to achieve bone health, brain health, heart health. And that all of the body of evidence is actually looking at that. So I don't think we have to do away with menopause for all the reasons that were laid out, but rather just achieve that goal. And to answer that question, I think that really needs to be a major focus for investment. Our conventional women's health channels and strategies around drug discovery and treatment is entirely focused on delivery of babies and surgical removal of pathology and not actually helping women age well. So women do have these biologic predictors of longevity. We also have unique susceptibilities. And if we can hone our understanding of the female hallmarks of aging, I would expand on the ovarian hallmarks that you showed, just these incredible sort of markers of successful aging and how we are more susceptible throughout our reproductive lifespan and beyond with unique targets for intervention.
Thank you, Jennifer.
Yeah.
And to build on that, based on what I spoke about today, I think if we had a way to look at human ovarian tissue, I think that would really be a game changer. It's an ethical dilemma, but I think there's more to the ovary than we really know, particularly say in young girls. I think testosterone has a bigger role and they need to be treated differently. So,
we have a dramatic entry that signals that we are over time, way over time. Okay, maybe to close that up, please reach out to our speakers after the coffee break, ask them all the questions. One big round of applause for everyone. And